what we're going to do is do a quick switch and have David Segal present his work from the FIRE team. David is the Associate Director of Genomics at UC Davis Genome Center, and he's an Associate Professor in the Department of Biochemistry and Molecular Medicine at the School of Medicine at the M.I.N.D Institute. So that should sound really good. So Dave is going to be talking about some of the work he's been doing that's really exciting on molecular therapies in Angelman syndrome.
Thank you. Thank you. Thank you. Hi, I'm Ann Anderson, and no, I'm just kidding. When we train graduate students to try to keep up with all the literature, oftentimes we'll have what we call a journal club, where we'll meet and we'll talk about articles that are out and we'll try to discuss them. And so really what I wanted to do with you today was have a journal club about molecular therapies that many of you may have heard about and know something about. And I'll try to set the record straight, but of course it's setting it straight from my perspective. So some things I want to talk to you about are what are gene transfer, what's a topoisomerase inhibitor, what's an artificial transcription factor, and what's an antisense oligonucleotide. We're going to talk about how these can be applied to address the underlying causes of Angelman syndrome. And then I want to close with some issues that need to be addressed, I think, before these can advance to clinical trial, any of these. So I think everyone in this room is kind of familiar with this kind of slide about the kind of genetic mutations that give rise to Angelman syndrome. So often there's a large deletion on the maternal chromosome. So that region of the maternal chromosome is completely missing. Of course, there may also be some point mutations. Do I have a pointer? Okay. Point mutations in the maternal copy of UBE3A, or some uniparental disomy, or there's some other imprinting defects, some other kinds of things. From studies that have come up, we know that let's say at some level, the underlying cause of Angelman is loss of UBE3A expression in the brain. And so a kind of molecular strategy, or a goal of the molecular strategies, is to try to restore UBE3A expression in the brain in some way. So in the stories that I'm going to tell you about, there's actually two mouse models that keep coming up. So I just want to introduce them so that everybody's clear on that. One is the so-called YFP mouse. And the YFP mouse has a fluorescent tag, if you will, that's in front of this UBE3A gene. So this is the UBE3A gene. This is the one on the maternal copy that's normally on. This is the one that's on the paternal chromosome, and that UBE3A is normally off. Here, Scott Dindot actually attached a fluorescent protein to that when he was in Art Baudet's lab. And the consequence of having that fluorescent protein there, so normally that gene is off and that fluorescence is not on, but anything that activates that now makes a fluorescent signal. And so this mouse strain is very useful in screening things that might be able to turn on UBE3A. So people use this, for example, for drug screening to try to find drugs that could activate UBE3A. It's important to note, though, that this is not a model for Angelman syndrome, because the maternal copy of UBE3A is there and intact. So the other model that is often used in this research, we'll just call it the Angelman syndrome mouse model. And that also does not have the large deletion that is often found in humans, but instead it has a disruption of one exon, one part of UBE3A. And so it might be something like a point mutation. It's a little bit larger, the mutation's a little bit larger than that, but it's important to consider it's not that whole region when we talk about this Angelman syndrome mouse. And both of these were made in Art Baudet's lab. Okay, so you have a gene that's not on in the brain anymore, and you would like to have it on. And this is a very kind of traditional goal of gene therapy in general, or let's just call it gene transfer therapy to make it very clear what the therapeutic goal is. And anytime you have on a very simple level, if you have a gene that has a mutation and it doesn't work anymore, and you need that gene to do its job in the cell and gives you a disease if it doesn't do that. The simple idea behind gene therapy is that you just introduce a healthy copy of that gene into that cell, and whatever that thing used to be doing, now this thing will do it, and you should be good. So, so the first study I want to talk about is actually one of Ed Weber's studies. It's always kind of funny to talk about this while the author's in the room. So hopefully I don't say anything too stupid, but it's really very interesting. And actually, this paper I think really set the groundwork for all the other studies that came after it. And so to introduce this gene, they used a virus, which was an adeno-associated virus. It's just a viral vector, and the virus it's not like a infectious virus that gives you a disease, but it's kind of been retooled for biotechnology purposes to carry the UBE3A gene. So this virus is going to take the UBE3A gene, bring it into the cells in the brain, and it's going to sit there and express itself. And so when they treat the Angelman Syndrome mouse model with this viral vector, they can deliver UBE3A into the brain. So this is a wild-type mouse, and this is a region of the brain in the hippocampus. And you can see this kind of dark staining pattern. You don't see that dark staining pattern in the Angelman Syndrome mouse model, but when they put in their adeno-associated virus, you see the stain again. So that means that region's making UBE3A again. Great. So what does that do to the mouse? So fortunately, Dr. Weber has already explained all these assays. But I would just state that these are the kinds of things that they reported seeing in the mouse. So one of the assays was a learning assay. This is the contextual fear conditioning. And as Ed just pointed out a little earlier, in the wild-type mouse, they have some kind of a signal that's up here. And in an Angelman syndrome mouse that's treated with a kind of control vector, the learning of that mouse is less. But when they treated with this UBE3A from this viral vector, it goes back up. So basically, they were able to rescue this learning phenotype in the mouse. This is also this long-term potentiation study that he introduced as well. And again, all I want you to remember about that is it's basically a kind of study of how well the neurons are working with each other. And all you need to see in this is this blue line is a wild-type mouse, and it's doing pretty well. This green one is an Angelman syndrome mouse treated with a control protein, and it's not doing as well. And then the one treated with UBE3A, the red line is closer to that blue line than it is to the green line. So that's all you have to know, is that the treated mouse looks more like the wild-type mouse than the Angelman mouse. So that's good. Now, one of the concerns about using viruses in the brain to deliver genes, however, is that sometimes they have a kind of limited distribution. So in these experiments, they injected this adeno-associated virus into this hippocampus region of the brain, and that's where we saw all these nice results. And this is the center for learning that was described earlier. But cerebellum is another region of the brain that's often involved in motor function, and the way this was delivered in this case, the gene did not make it into the cerebellum. And so they could analyze this by looking at this. They can analyze the motor function using this rotor rod test that was also just described earlier. And again, in this case, you could see that they were not able to rescue the motor function. And again, all I just want you to see is that this blue line is the wild-type mouse doing pretty well. The green line is the mouse that only received the control, the Angelman mouse that received the control, and the red line is now down there instead of up here. So it didn't work too well. But we can understand that because the virus didn't get over there, okay? Now, I'm telling you all this because I want you to remember these graphs, because we're going to see them again in a little bit. So, summary of this kind of gene targeting approach that's been used. It did turn UBE3A up, so that's great. It fixed some of the symptoms like learning and neuron function. But the distribution is limited in this approach, so it didn't get into the cerebellum, and therefore we didn't see a rescue of motor function. Now, I know that Ed has some great ideas about how to improve upon this kind of approach, and we'll be very excited to hear how that goes. Now, Ben Philpott's lab took a different approach, and they were interested in screening drugs that could possibly turn on UBE3A. And I have to say that I would have been very skeptical about, I am still very skeptical about this approach because, you know, what I know about drugs is that drugs like to bind to small hydrophobic pockets on proteins. So they're good for blocking an ion channel, they're good for blocking a catalytic center of a protein or an enzyme. We don't really have drugs that can stop something from binding to DNA or turn on a gene, you know, by binding to the DNA or something like that. But nonetheless, Ben Philpott's lab used the YFP mouse. Remember, I told you that was a good model for seeing if you could turn this gene on. And they just used cells from that YFP mouse. And they screened over 2,000 compounds. And all you need to know from this graph is that things that are above the line turn the gene on. And the ones that are in gray here are actually what they call false positives. That mean that in their screen looked good, but when they went back and tested it, it didn't actually work. There was one drug that they tested out of all the 2,000 whatever. There's one drug that seemed like it turned it on. And that was this drug, irinotecan. And this is a topoisomerase inhibitor. And I think it was very surprising that that class of molecule was able to turn on this gene and turn it on so well. Now, this is a drug that, that doesn't cross the blood-brain barrier. So in us and in mice, there's a barrier that separates the normal blood that goes through most of our body from the brain. And it's thought that this is something that evolved to kind of protect the brain from stuff that might be circulating in the blood. So unlike aspirin that you could just take orally and then it gets all over, you know, up into your brain too, in this case they had to introduce it with an injection, and the injection was with a pump here. So they had to pump it into this kind of ventricle space into the brain, and when they did that, they could see that it got into, so if they pumped it into this side of the brain, they got it into this part of the hippocampus, and it didn't get so much in this other side of the hippocampus, so they could compare those things and see. But you can see, you know, that UBE3A, so UBE3A is now this green stain, and it's coming on. And so that's good. It's definitely coming on, and they can quantitate that, and you could see when they add this drug, now it's a different version of the drug topotecan, that they get a big increase in the amount of UBE3A that comes on. Interestingly, if they just wait 5 hours later, all the drug's gone. So they put it in, it goes boom, and then it goes boom. Now, how could it be that a topoisomerase inhibitor is working like this? So I just want to try to give you an idea of what this drug is actually doing, because it's important to consider this for what it can do and what it can't do. So, DNA is a double-stranded molecule, right? It has that double helix, so there's two strands of DNA that are wrapped in a double helix. And that double helix is wrapped up like two strands of a rope wrapped around each other, okay? And now, when a biological process, like when you go from DNA to RNA, this process of transcription, this takes proteins that need to open up those strands of DNA and read one of them and turn one of those DNA strands into RNA. So now we're going to have a protein running right down the middle of those two strands of this double helix. Okay, so you can imagine if you had I wish I had a rope, but if you had, two pieces of rope that are kind of wrapped around each other, okay, and you take a stick and stick it between them and shove it to the end, all those turns are going to get shoved to the end, and it's going to kind of make a knot. And that's essentially what's happening in DNA. So, so in green is the enzyme that's going to read this DNA and turn it into RNA. That will eventually go on to make protein in many cases. And what's shown over here is that as this thing moves along, it builds up these twists in front of it, kind of unwinds it behind it. And so eventually, it's going to run into these knots, and it's not going to be able to go forward. So imagine, again, with the rope, if you had someone on the other end of the rope unturning it as you were going down there, then you'd be able to proceed. And that's what topoisomerase does. It kind of unwinds these knots as this enzyme is moving down the DNA. And so it's important in this process of converting DNA into RNA. And so normally we have topoisomerase doing that. When you inhibit topoisomerase from doing this job, you get these knots, and now this enzyme is not able to get all the way down. And so the way that this works kind of over here, the way we can think about it is an RNA molecule is going to start to be made over here. It's going, it's going, it's going. It's running into some knots. And where's my topoisomerase? I need it. I don't have it. So it kind of stops. And when this thing stops, this RNA doesn't go all the way to the ends. It doesn't act as an antisense to this UBE3A, and now this gene is able to come on. And so this was a very important study for the whole field, because it was really one of the first pieces of very hard evidence that if you could just stop this strange antisense from being made, or at least from going all the way to the end, then UBE3A will come on by itself. And so this kind of gave proof that there's this other kind of strategy. Instead of adding the gene in with a virus or something, what if you could just activate the gene that's already there, but it's silenced? So this looked very promising. But think about that mechanism that I was talking about, where the strands just kind of get tied up into knots during this process. It turns out that the longer the gene is, the further that RNA has to go when it's being made. It runs into this kind of knot problem, and it can't go forward. And unfortunately, this antisense transcript is not the only big gene that's in the brain. There's a lot of other genes that are very long, and the longer they are, the more they need that topoisomerase. So when you inhibit topoisomerase, it inhibits those genes being turned into proteins as well. So this is just a graph that says for genes that are longer than about this size of 100,000 base pairs, they start to become inhibited with this topoisomerase drug as well. So there's a lot of things that they can do with these topoisomerase drugs that medicinal chemists can help make them longer lasting, make them get into the brain better, maybe make them go across the blood-brain barrier. They can fix all those things. But one thing that they're going to have trouble fixing is if the mechanism of this drug is to inhibit topoisomerase, they're going to run into this problem that they saw about 155 other genes being silenced from this action. So, summary of topoisomerase inhibitors. It did turn UBE3A up, and it did that because it turned that antisense transcript down. So, that's a really important discovery. Having a drug is good because, we know a lot about drugs. People could take drugs. This one can't be swallowed because it doesn't cross the blood-brain barrier, so it'd have to be delivered with some kind of pump. The action, I think, has been too fast to examine symptoms in a mouse model yet. We might see this coming at some point, but at this point, I'm not aware that they've reported, in the literature at least, rescue of a phenotype with this drug. And then there's other genes that get turned off by this treatment just because of the action of this drug. That doesn't mean that all drugs will have that kind of mechanism and those kinds of problems. But that particular class of drugs had that problem. So another way that we could turn that turn that antisense down and then hopefully reactivate the UBE3A is with transcription factors. So this is how nature does it, right? If nature wants to turn a gene on or turn a gene off, it uses these proteins called transcription factors. And so transcription factors are things in our cells, and they typically consist of two different parts. One is a DNA binding domain that recognizes a specific sequence of DNA. So that's going to bring this transcription factor to the UBE3A gene, to the SNRPN gene, to the ARC gene, whatever. And then it has this other part that we call an effector domain. And that can either help to turn a gene on, or another kind of effector domain can turn the gene off, depending on what this thing actually is. And it does that with interacting with other proteins in the cell, blah, blah, blah. But to make a transcription factor, you just need a DNA binding domain, and we steal an effector domain off of a naturally occurring transcription factor. So if we could make artificial transcription factors that could either activate UBE3A or turn off this antisense, we should be able to get that UBE3A gene activated. And so that was the kind of approach that we were taking. So the results I'm going to show you about now, and this is our study. I couldn't start with a paper because we don't have the paper yet. But so we're going to make artificial transcription factors, and the one I'm going to tell you about is actually a repression factor, and it's going to repress this antisense transcript. So I like to think we're doing something a little closer to how nature would do it. And right, so we're going to make this molecule, and it's going to go and bind right over there and hopefully shut that off. Now, what about that DNA binding domain? So now there's several different kind of platforms for trying to make engineered DNA binding domains. And all of these things, zinc fingers, something called tau proteins, and this very new one, this CRISPR kind of protein. All of these things are proteins that can be engineered in some way that you can target them to a specific site in the DNA. I'm glossing over about 20 years of research to bring us to that point, but let's say these things can be made now, and then again we can pull off a repression domain off of naturally occurring transcription factors and stick it onto those, and so now we can target this repression activity anywhere we want, and we are going to use these things to target, to turn off that antisense transcript. For various reasons that I'm not going to go into now, I'm going to show you the results that we have from the zinc finger study. So we made this zinc finger protein with a repression domain attached, and a bunch of other stuff as well for various purposes. And one of the things that we stuck onto it is this so-called cell-penetrating peptide. And this is a little bit of a protein that endows the rest of the protein with the ability to cross cell membranes. And so this is going to be our way of getting this into the cells. So we can just make this purified protein and inject it into the mouse. And because it contains this cell-penetrating peptide, it can cross cell membranes and get into the neurons in the brain. So when we inject it into the mice, and we look to see, is this protein getting into the brain? In fact, it is getting into the brain, and we were very happy to see that it seems to be getting in all over the brain. So we have very widespread distribution of our artificial transcription factor in the brain. And I think that this is a real plus of this approach. I didn't know it was going to work this well in the beginning, but I'm very happy that it did. So it seems to really be distributed in the brains of treated mice. Of course, we don't see the signal in non-treated mice. And again, I would just contrast that with viral methods, where oftentimes you see the virus kind of near the site of injection, but not in distributing out very far from that. So this looks like something that's very promising. And the other thing that we measured with this is the time course of when we see the peak signal after we give a treatment. And we see a peak of signal in the brain at around 4 hours from our treatment. Okay, so if you remember the topotecan drug, it was completely gone by 5 hours. We're just reaching our peak at 5 hours, just to put it in perspective. And like the other treatments, this also turns on UBE3A. And again, since we see that widespread distribution, we wanted to look at 2 different regions of the brain. So this is the region where the hippocampus is. This is the region in the cerebellum. And whether it's hippocampus and cerebellum, this is the Angelman Syndrome mouse model. It's the wild type mouse, showing a lot of UBE3A expression. This is a protein that's kind of like our protein, but it doesn't bind in that region. And this is our therapeutic protein. And again, you could just see that there's more UBE3A expressed here. It's more like wild type than it is like the Angelman mouse. And then there's just some quantitation. So we're turning on UBE3A as well. And so now we could ask the question, what kind of behavioral changes does that give? But unlike the talk you just heard, in our hands, we were not able to get a lot of convincing phenotypes from this mouse model. So we did see some phenotypes just looking at wild type and Angelman syndrome mice with the rotor rod and some of these motor kinds of tests. And we also saw, things like marble burying, which, I don't really know what to think about the marble burying phenotype. Some people use that in their publications. But for us, the mouse model didn't seem like it was, the current mouse model that we have didn't seem like it was a very robust place to test our therapeutic. So with the new genome engineering tools that are available, we started thinking about better mouse models. And so we're in the process now of creating a mouse that's going to have a larger deletion than the deletion that is than the disruption that's currently what we refer to as the Angelman syndrome mouse model. And we think that will be very helpful. Hopefully we'll have more profound phenotypes, more like people, and we can use that. As was mentioned also, we're interested in a rat model because the rat model is a better model of cognitive ability. It's basically a better model for almost anything that we want to look at for Angelman syndrome. So that's the impetus behind the rat model. Now, why didn't we use the rat model years ago? Because we couldn't. The tools that were available, there were robust tools available for making genetically modified mice. But those methods didn't work in the rat. So nobody could make a mouse model. It's just within the last 2 years that we've actually had this ability, we as a community. And so now we see more mouse models and rat models coming online. And so we want to have a rat model as well because it's going to tell us more than the mouse model was able to tell us. And then the pig model has already been described. And so all of these new animal models are possible because of these genome engineering tools. So hopefully when we get a better model, we'll be able to also study the effects of what this does on the behaviors of the mice. So in summary, for artificial transcription factors, it also upregulates UB3A because it's downregulating this antisense transcript. We have widespread distribution in the brain. We're concerned that in our hands, the AS mouse is not a good enough model to examine symptoms, and we're not published yet, but we should be soon. The last one that I want to tell you about are the so-called antisense oligonucleotides. And this is something that was published just last week in Art Baudet's lab, although he's talked about the data a little bit before. And so the antisense molecule is another little stretch of nucleic acid. And it basically is designed to bind to the piece of RNA, in this case, that's being made at this gene. So the antisense molecule is usually some kind of a DNA that's designed to bind to a piece of RNA. And they're very easy to design because you know the nucleotide sequence, and you can just code it, and it's very easy to synthesize it. When they use it as a therapeutic, they dress it up a little bit more with some medicinal chemistry tricks. But basically, that's what it does. It's a little strand of DNA, and it's going to bind with a very, specific interaction with this RNA. Now, who cares about that? Why would we care? So it turns out that if you take one of these antisense oligos, and bind it to a piece of RNA, there are proteins in the cell that degrade RNA DNA duplexes. Why they're in there is a story for another lecture, but we have these proteins in our cells, and so anytime you can make a little short strand of DNA that binds to the RNA, this enzyme will come along and chop it up. So the effect of having this antisense there is that this RNA is going to be chopped up right here, right where that thing is targeted to. And so, in what they were hoping for was that, kind of like the other strategies that are disrupting this antisense, if they chop it off right around here, it won't, proceed into this region, or it won't interact with this region in some way, because this end is going to get chopped off, and now this UBE3A gene is free to come on. And in fact, they were able to show that it does come on. This is in the YFP mouse again. And so here's a control mouse. They can show that their antisense oligonucleotide gets into it, but they don't get that signal that's created when the UBE3A turns on. But when they put in this therapeutic antisense molecule, now they see that signal come on, meaning that UBE3A is being turned on. They did this in a very clever way. There's this antisense molecule starts way over here near the SNRPN gene, and it goes over here, all the way 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 over here, and all the way over here. It's one of the longest RNA molecules, I think, in the whole human genome. And as you probably know, there's other parts of this RNA molecule that get chopped out, and they have some other function. And in Prader-Willi individuals, we believe that a lot of the defects that they have are because they don't express this little cluster of RNA pieces. They don't get cut out of this molecule, so they don't have their SNORD116 RNA pieces coming out of this long transcript. So by designing the antisense oligos over in this region, they reasoned that they could just eliminate the part that goes over UBE3A that acts as antisense to UBE3A, and all this stuff would be there. And that's exactly what they found. So again, looking at this graph, this just says that when they treat with these antisense oligos, the antisense transcript as measured over here goes down, UBE3A expression goes up, okay, but the SNORD116 expression doesn't change. So this part of the antisense is not made. Now UBE3A is able to come on, and all this stuff stays there. So all that seems very, very good. That's all very, very good. They also wanted to show that unlike the topoisomerase drugs, which we said inhibits all these long genes, they measured some other long genes, and they also showed that their antisense approach did not inhibit these long genes. So that's all very, very good too. So the antisense does not affect SNORD116 or these other long genes. And they also showed that this has a very long half-life, a long duration of action. So I don't want to explain all of this graph, but this is just the UBE3A coming on, and this is weeks after a single treatment. So this is weeks after a single treatment. They add this antisense oligo into the brains of the mice, and up to 16 weeks after treatment, that's like 4 months, they still see UBE3A on from that one injection of this antisense oligo, and then it starts to come down. So basically, a single treatment with this antisense oligo seems to last for 4 to 5 months. That's also very, very good. Now, like our artificial transcription factor, they also showed that they had very widespread distribution in the brain. So that's also very good. And then, like this viral approach that I mentioned earlier, they also did some this contextual fear conditioning, or this learning assay. And I thought this was very interesting. Okay, So this is Ed Weber's data that they published in their paper. See, it's wild type is high, the sham-treated animal is, Angelman syndrome animal is low, and then the introducing UBE3A brings it back up. Now this is the same kind of thing with their antisense, and look at that. I mean, doesn't this up, down, up look exactly like this? I mean, these are different, different mice, different studies, different treatments, and this data set really looks very similar to this one. In the same kind of assay. So it's giving the same kind of rescue. So that's great. They also looked at this body weight that was mentioned a little earlier ago. Although I have to say that it seems that this change in body weight was mostly due to an effect that happened really in the very early time when there was a dramatic weight loss of these Angelman mice. And then they kind of like continue on up in a way. So I'm still trying to understand what's going on with that. But anyway, those are things that they showed were definitely working as a result from this antisense treatment. They also looked at motor function using that rotor rod test. And in fact, they were not able to rescue the rotor rod test. But I just want to show you this again. Okay, this is the graph from the viral study. And this is the graph from the ASO study. You see how similar they are? I mean, isn't that freaky? I mean, it's exactly like the same curve. And this is now, different, same mouse model, but, years apart, different treatments, different labs, and these curves look almost exactly the same. So I just thought that was pretty impressive. But so they were able to rescue some phenotypes like learning, but not able to rescue other phenotypes like the motor function. But in this case, in the viral case, we know that's because the virus didn't bring the UBE3A gene to that place. The question is, why doesn't this one rescue the motor phenotype? Because supposedly this was distributed brain-wide. So I think we have a lot to learn about what these ASOs are doing and how they're doing it. So let's say it's early days, but it's a very encouraging early day. So again, in summary, UBE3A is going up with this treatment because the little bit at the end of UBE3 antisense is going down. It has very widespread distribution in the brain. One treatment lasts for 4 to 5 months. We don't see silencing of long RNAs or SNORD116, and it fixed some symptoms, but not some other symptoms. So in the last couple of slides, I just want to end with a paper we actually did publish, where we just talked about the prospectus of these molecular therapies, and we said that we proposed 3 main issues that we thought need to be addressed to advance these towards clinical trials, and they would be specificity, toxicity, and delivery. So let's just see how our contestants stack up in those categories. Okay, so there's the viral approach, the topotecan, topoisomerase inhibitor, the transcription factor, and the ASOs. So delivery is probably one of the hardest issues of any kind of therapy, even drug therapy. For both the virus and this particular drug, they're not able to cross the blood-brain barrier. So we're looking at either intracranial injection or intrathecal injection. So intrathecal injection is injecting into the cerebral spinal fluid in the spinal column. And one of those 2 methods would be the route of administration, particularly for, maybe for the drug. And certainly that's something that Ben Philpott has been exploring. In addition, the viral delivery method has a distribution that localizes it kind of near the point of injection. But like I say, there may be ways that a variation of this might be able to get around that. The artificial transcription factor seems to work well. Even if we inject it into the belly of the mouse, it's still able to cross the blood-brain barrier and get into the neurons in the brain. And it shows very good distribution when it's within the brain. So that delivery mechanism seems to be working surprisingly well. Now, these antisense oligos are also not able to cross the blood-brain barrier, so they also have to be delivered with this kind of intracranial or intrathecal injection. But it has very good distribution in the brain. I added in duration. It wasn't one of my original challenges, but I just thought I'd make that clear. This gene therapy would be something like a long-term approach. I want to be careful not to use the word cure, but let's say it would have a very long-term effect because it would be fairly stable and stay for a long time, make UBE3A after one treatment. Now that's kind of a plus and a minus because as a researcher, we think that's where you want to go, right? You want to like treat them once and then they're done. The clinicians that I work with have a very different attitude about that sometimes because they always worry about side effects. And if you do something that's permanent, they get nervous that, how do you stop it? So they like things that have a transient effect more often. The whole field is kind of hashing this out, but let's say we would expect that this approach would have a long-term effect. The drug was very transient, so it would need almost continuous dosing. But again, with some medicinal chemistry, maybe they could improve that. This transcription factor was also transient, although a little bit longer. It would still need periodic dosing, and I could tell you in our lab, we're working on other methods so that we have a more persistent effect, something closer to that. This one was transient, but very long-lasting, many months, so an individual might need something like 4 or 5 doses per year. This is Art Baudet's own suggestion, is that the treatment might require like 4 to 5 doses per year. Toxicity, adeno-associated virus is generally not toxic in the brain. We're not getting endogenous gene regulation, but we don't know if that's important in this effect. Certainly was able to rescue some phenotypes. This topotecan drug is, in its other life, is an anti-cancer drug that makes strand breaks. So Ben showed that at the doses that they use, they don't see that kind of toxicity, but their newer data shows they have other kinds of toxic effects. This protein, because we're injecting this protein, and right now we have to inject it a lot, we might eventually see an immune response against the protein, and that's something that we're studying carefully. As far as we know, these antisense oligos are well tolerated, and the company that Art Baudet works with has a lot of experience with these things. So that antisense is a winner there. Specificity, the virus is very specific because it only makes UBE3A, and so that's as specific as that gets. This drug, again, has some, you could consider them off-target effects that it inhibits all these other genes. This transcription factor might have some off-target effects, and we're looking into that now. It might affect some other genes. We haven't seen evidence of that, but we're looking more carefully now. And this one, I think we don't really know. It shouldn't have any off-target effects, but we don't know. I don't know if we know that yet. So we have a lot to learn about this. But I think that the other thing that I didn't put on all these slides that is really the message that you should all leave with today, optimistically, is that I'm showing you a slide here of 4 different things that can turn on UBE3A in the brain. So just think about that, where we are. I mean, 2 or 3 years ago, none of this stuff was here, And today, we're talking about 4 different kinds of molecular therapies that have been shown to turn on UBE3A, at least in the brains of mice. So, I mean, just think about where we are with that. And I hope that leaves you with a sense of hope in the future. And in closing, I want to thank you for your attention. And I also want to thank you all for your support of this research. This is the plaque, actually, that is hanging outside our imaging facility room. And a special thanks to the family of Jamie Berkeley for giving us the ability to do this great work, and all the images that I showed today are the result of that contribution. And I also want to thank the other members of the FAST fire team and FAST in general, and we've also had some support from the National Institutes of Health. So I'll stop there and take any questions. Thank you.
So we have time for 1 or 2 questions, and then we're going to take about a 10-minute break between, because I know you're probably all thinking, wow, I had lunch and you just made me sit here for 2 hours. There was a question in the back, and I'll come get that one. So we'll do two. Why don't you do this first one, and then...
In the ASO research, I was just curious if it were ever to go to humans, do you think that I know you didn't experience much or any motor function improvement, but is it possible that in a higher order animal, human, that increased cognition could result?
Seizure, reduce those chemical reactions. I know, except the problems, genetic problems, is there any outside factors like toxins or anything that can actually increase the risk of seizures, so we can avoid those factors and just reduce those seizures and help all children? That's one thing. And then the other thing is that's great study to help the activate the UBE38. There are some cases when there is a mutation of that chromosome. So I was wondering if there's any way to, any studies, like to use umbilical cord from the sibling in order to replicate, to clone those pets UBE3As.
So to address the first question about exposures and does that have something to do with seizures, I mean, I think you've heard the state-of-the-art seizure, where we are with seizures already this morning, and I would defer you to our seizure experts, but I would just say that certainly there's a lot of issues that we need to address with any kind of therapy. And whether the kinds of things that I talked about will be able to address seizures is still a very open question as well. And so that's also something that we have to look at. As far as what's causing the seizures, yeah, that's a whole nother subject. As to your second question, you know, could there be something genetically that we could do to fix the genetic defect that's leading to this disease? Someday there may be, but today there's not. We just, as a research community, we just don't have the tools and the know-how to be able to go in and make a genetic change in all the neurons of the brain. Maybe someday we will, though.
Okay, if you have other questions for David, you can save them for the last Q&A session. Let's thank David, and we're going to take a quick 10-minute break.